Hi everyone, it's Dr. LeBlanc with a video capture lecture on prokaryotic transcription. The learning goals for this lecture are to learn how RNA is synthesized from a DNA template in the process of transcription, what the three phases of RNA polymerization reaction are, what a promoter is, and its structure-function relationship with the gene, what the basic structure of a gene is and how it relates to the structure of a transcript, how RNA polymerase and its subunits function, how proteins bind the DNA, how sigma factor directs the transcription of specific genes, what the transition from a closed to open complex of DNA and RNA polymerase means for the initiation of transcription, what the template and coding strands are, and what row-dependent and independent termination are. This is a model of RNA polymerase. It's a space-filling model here with the blue and gray subunits. Here's DNA coming into the RNA polymerase molecule. This is the space-filling portion, and this is the backbone model. Right here, the two strands are separated, and you can see an RNA transcript in red being produced. A gene is a segment of DNA used to make a functional product. Transcription is the process of copying DNA into RNA. This diagram illustrates the major steps of the central dogma. The process of making more DNA from DNA that you already have is called replication. The process of going from DNA to RNA is the topic of this lecture, and this is called transcription. Later on, in subsequent lectures, we're going to learn about the process of making a protein from RNA, and this process is called translation. Now, in order to keep these straight, it's sometimes easier to think of translation first and then remember that the other process is transcription. Translation is going from a nucleic acid to an amino acid. It's a completely different molecule. Transcription is when you go from one type of nucleic acid, DNA, to another type of nucleic acid, RNA. Okay, so transcription is what we are concerned with in this lecture. But this diagram again indicates the central dogma of DNA makes RNA makes protein. RNA transcripts have two major types of function. 90% are known as structural genes. They encode messenger RNAs that go on to produce proteins. In addition, approximately 10% of genes that encode RNAs are untranslated RNAs. In other words, they are not messenger RNAs. They are not translated into proteins. Instead, they function simply as an RNA molecule. They are things like ribosomal RNA, tRNAs, microRNAs, SNO-RNAs, which are important in ribosomal RNA processing, snRNAs, which are important in messenger RNA processing, and a whole bunch of others, and we keep finding new ones all the time. This diagram indicates that a DNA gene has segments that surround a gene of specific nucleotides, and that the messenger RNA transcript, this is a messenger RNA transcript down below here, has sequences of nucleotides that function as RNA. Okay, so this is one gene here. It stretches from regulatory sequences at the 5' end of the gene. It includes a promoter region, which is what we are going to be considering in detail in the rest of this lecture. The promoter. The promoter is a sequence of nucleotides to which RNA polymerase will bind. I'm just going to abbreviate this RNA polymerase, Pol. Okay, so a molecule of RNA polymerase will bind here to the sequences of DNA, the nucleotide sequence in the promoter region. At the end of the gene, we have terminator sequences. RNA polymerase will bind here and it will fall off here. After we've produced a messenger RNA, some of the sequences that function as RNA are the ribosome binding site, the start codon, the many codons that encode all the amino acids of the protein, and finally the stop codon. We'll talk in a lot more details about this in subsequent lectures, but I just want to indicate that the nucleotides that comprise the ribosome binding site are encoded in the DNA of the gene. And you can map the ribosome binding site of the messenger RNA onto the gene. So similarly, you can do with the start codon, all the codons, and the stop codon. So you can find the nucleotide sequences for the start codon and all the rest of the codons, including the stop codon, encoded in the DNA of the gene. There are 3 stages of polymerization reaction. Similarly to the stages of DNA synthesis or replication, we have initiation, we have elongation, and termination. Initiation in prokaryotes involves RNA polymerase binding to a promoter, and this is a promoter, a typical promoter for a prokaryotic gene. Let's orient ourselves. +1 nucleotide is the first nucleotide that is transcribed into RNA. Okay? The promoter region is the upstream region. We call this region upstream, and this is downstream in this direction. So the promoter region is upstream of the start site, and it has 3 main regions to it. This is the transcriptional start site here. The bases to the right are labeled with a positive number, and to the left are labeled with a negative number. There is a -10 sequence, which means that it is 10 nucleotides away from the start site of transcription, the first nucleotide. And notice that it is quite AT-rich. There's a -35 sequence, which is not so AT-rich. It's a mix of nucleotides. And then there's a region in between that's from 16 to 18 base pairs. The sequence of nucleotides can be any sequence. There is no specific sequence of nucleotides here. But what is important is that the number of nucleotides remains between 16 to 18 base pairs. And why is that? The reason that this has to be 16 to 18 base pairs is because there's a factor called sigma factor that binds to both the -10 and the -35 sequences. This is a diagram of a consensus sequence looking at the promoter regions of several different genes. Notice that the -10 is very AT-rich, and the -35 is are different combinations of nucleotides. And the space in between the -10 and the -35 is from 16 to 17 nucleotides long. If we look at the individual positions of each of these nucleotides, we find that for the first position, T is the most common nucleotide. The second position is usually occupied by an A, etc., etc. Notice that none of these, well, except for this one, only a few of these, are exactly the same as the consensus sequence. This is the Rna gene. And the consensus sequence for the -35 region here, it's the same as the trip operon. This is a diagram of RNA polymerase in red with its associated sigma factor. Notice that sigma factor binds to both the -10 and the -35 region at the same time. And when sigma factor is bound, RNA polymerase can bind as well. In E. coli, RNA polymerase holoenzyme is comprised of a core enzyme with the two alpha subunits, a beta and a beta prime subunit, and an omega subunit, which you can't see because it's on the other side of the two beta subunits. It also has sigma factor bound to the -10 and the -35 region. This is it right here, binding tightly to the promoter region. The direction of transcription is going to be from left to right, so when RNA polymerase and sigma factor are bound, the RNA polymerase is positioned in exactly the right spot so it can begin to transcribe RNA at position +1 of the gene. This is a diagram of how the sigma factor is binding to the DNA double helix. Notice that it contains a very classic motif for a DNA binding protein. DNA binding proteins often have this alpha helix turn helix motif, a helix turn helix motif, where the two helices bind to opposite sides of the DNA in the major groove. The sequence of amino acids on these alpha helices interact with the sequence of bases that are protruding into the major groove of the DNA molecule, so that this interaction of the alpha helix and the DNA is very specific. The transition from an open complex, a closed to open complex, is the two phases of initiation of transcription in prokaryotes. RNA polymerase with sigma factor on it will scan along a DNA molecule until it encounters a promoter. After sliding along the DNA, the sigma factor recognizes the promoter, and RNA polymerase holoenzyme forms a closed complex. Here it is bound tightly to the DNA at this position, with sigma factor interacting with the -10 and the -35 and RNA polymerase holding on to the DNA. Notice that in the closed complex, the DNA remains hydrogen bonded. The two strands of the DNA molecule remain hydrogen bonded when RNA polymerase is attached to the DNA. The promoter escape and the start of elongation occur in a transition from a closed complex to an open complex. RNA polymerase often starts and stops and starts and stops during that promoter escape transition. When it finally produces an RNA transcript of approximately 9 nucleotides long, then sigma factor falls off, the open complex remains consistent, and RNA polymerase can proceed down the DNA through the gene, producing an RNA transcript. If sigma factor remains associated with this, the promoter does not, the RNA polymerase does not escape the promoter. It remains bound to the promoter. So in order for RNA polymerase to escape the promoter, to leave the promoter, it must lose sigma factor. Okay, if we look up close and personal at the catalytic portion of the RNA polymerase, we see that in the catalytic part of the enzyme, the DNA strands are separated, and RNA is actually base paired to one of the two strands of the DNA in the transcription bubble. That's what this is called, the transcription bubble. So here's the direction of transcription. We are adding nucleotides to the 3' end of the transcript. Here is the 5' end of the transcript. RNA polymerase has a couple of very interesting features. It unwinds the DNA upstream, and downstream it will rewind the DNA back again. If we look at this transcription bubble up here, we notice that we have labels for the two DNA strands. One of the two DNA strands is called the coding strand. It contains nucleotide sequence that's the same as the RNA transcript. The purple strand here is called the template strand. The nucleotides of the template strand are complementary to the RNA. The template strand actually is doing the work here. It is the strand to which the incoming RNA nucleotides hydrogen bond, and RNA is actually synthesized off of the template strand. This phase is called elongation. So this will proceed for as long as it takes to finish transcribing the entire gene. This is another diagram showing that the coding strand has the same sequence as the RNA, and the template strand is the strand to which the new nucleotides coming in are hydrogen bonded as the new phosphodiester bond is made at the growing 3' end of the transcript. If we look at a genomic view of this, on a chromosome, we can have genes that are placed in either direction. Gene A here and gene B both have their promoters on the left-hand side. Gene C has its promoter on the right-hand side. Both gene A and gene B will use the bottom strand as the template strand and synthesize RNA by hydrogen bonding to the template strand. So RNA is made in a 5' to 3' direction off of the template strand. For gene C, we have a promoter that is on the right-hand side. So here, the direction of transcription is from right to left. The 5' end is on the right and the 3' end is on the left. So the other strand of the DNA molecule for this gene is the template strand. There are two different types of termination found in prokaryotes: row dependent and row independent. What's row? Well, this is a diagram, a ribbon diagram of row. It has 6 subunits, all nicely colored in different colors in this diagram. And notice that they form kind of a horseshoe or a clamp. Row binds to the messenger RNA, or any RNA, close to the end of the transcript at a site called RUT, or row utilization site. The RUT site functions when it is transcribed. It functions as an RNA. When RNA polymerase has transcribed the DNA and produced an RNA with the RUT sequence in it, the row protein clamps on. RNA polymerase proceeds until it reaches the terminator, when it forms a stem loop that causes RNA polymerase to pause. Here is the stem loop that forms. The stem loop, remember, has hydrogen bonded nucleotides here and a single-stranded region here. RNA polymerase pauses due to its interaction with the stem loop structure. When RNA polymerase pauses here, row, which can travel along the RNA, catches up to the open complex and separates the RNA and DNA hybrid, and everything comes apart and transcription is terminated. The second type of termination is called Rho independent because it does not require the presence of the Rho factor. In this case, a stem loop is formed. But following the stem loop, the sequence of RNA in this region of the transcript, right at the very end, is U-rich. It has a lot of uracil residues in it. Remember, in RNA we have U in place of T. When this U-rich region is produced, it's not able to hold the RNA-DNA hybrid together, and termination occurs and everything falls off. And here's what the transcript looks like. It has all these U's at the 3' end and the stem loop. And for some reason, the DNA and the RNA fall apart, probably because these have only 2 hydrogen bonds with the template, and the RNA polymerase falls off. I gave you a partial list of different RNAs at the beginning of this talk, but here is another list of many different types of RNAs that are found in the cell, and here's a whole variety of structures that they form. This is a whole laboratory manual just looking at different ways to manipulate RNA. Here are some types of RNA: messenger RNA, tRNA, rRNA, ribosomal scRNA, the RNA of RNase P. Here's another RNA that we talked about before, the RNA in telomerase, and there are many, 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 many other RNAs. So in summary, we learned that RNA polymerase is a large multi-subunit protein that copies DNA into RNA in a 5' to 3' direction. That RNA polymerase binds at the promoter region. The promoters are comprised of specific consensus sequences of nucleotides, the -10 and the -35 regions, to which RNA polymerase subunits and associated factors can bind. Other parts of the gene include the transcribed region and termination sequences. The parts of an mRNA can be mapped onto the transcribed regions of genes. Messenger RNA parts include ribosome binding sites, codons, the start and stop codons, and untranslated regions. Bacterial initiation includes the binding of RNA polymerase to the promoter region via sigma factor, and scanning and abortive transcription before the transition from the closed to the open complex. Transition from closed to open occurs as the transcription bubble is formed with the subsequent dissociation of sigma factor. Elongation proceeds as the transcription bubble progresses down the gene, and new nucleotides are added to the growing 3' end of the transcript. Rho-dependent termination occurs when RNA polymerase pauses due to its interaction with the stem loop structure at the 3' end. The Rho protein binds to the RUT site of the RNA, scans the RNA, catches up to the open complex, finally separating the RNA-DNA hybrid at the transcription bubble. Rho-independent termination occurs similarly, except that the RNA-DNA hybrid dissociates when RNA Pol encounters the U-rich sequence of the terminator. There are many different types of RNA in both prokaryotic and eukaryotic cells.